So our first presentation, I'd like to welcome DeJager and Megan Perez of LeTourneau University. The title of their talk is Comparative Analysis of Mycobacteriophage Minor Tail Protein, Carbohydrate Binding Module, Polymorphism, and Host Range. Please welcome them.
Oh, no, the pictures are messed up.
Good morning. It's early, so we're going to keep this as exciting as possible.
So I am Megan Perez, and this is DeJager, and we're from LeTourneau University. And today we'll be discussing carbohydrate binding modules found in the minor tail proteins of mycobacteriophages.
All right. Here's our table of contents, if you care. Here's our methods materials, if you care about that too. All right, actually, let's get into the interesting stuff. Now, we annotated bacteriophage Andes this last semester. Andes is in cluster N. And since it's in cluster N, its sequence is homologous with the rest of the cluster N phages for most of its genome. However, there are a couple of points where Andes matches phages from other clusters. Well, we're looking at a point where Andes matches phages from cluster P1. We believe this is a result of horizontal gene transfer between viruses, where two phages infect the same cell at the same time, and some genetic information is exchanged. This explains why some random homology occurs in the middle of Andes gene 20, a minor tail protein. Now, using HHPED, we found that the inserted region actually codes for the carbohydrate binding module of hyaluronate lyase. This is interesting because it's in a minor tail protein. If you think about bacteria, they're covered in carbohydrates. So we hypothesized that this module is actually responsible for binding to the host. What's interesting is that we also found a carbohydrate binding module in Xerxes 20, which is what Andes 20 would have looked like without any insertion. What's strange about this is that Xerxes 20 and Andes 20 have different sequences, but they still code for the exact same structure, the carbohydrate binding module, or CBM, of hyaluronate lyase. This was very strange to us because we thought you have different sequences, they should code for different structures, but this isn't the case here. There's a couple of causes you hypothesized. First of all, of conservative amino acid substitution, and second of all, you could have different primary structures but still create the same final tertiary or quaternary structure. So here we have the two CBMs aligned here. They're clearly distinct sequences. This led us to the conclusion that cluster N and P1 must have their own distinct sequences encoding their CBMs, and this recombinant event switched out Andes' original CBM for the CBM initially found in cluster P1. Now this led us to the question, how many other phages, how many other clusters contain this specific CBM?
So let's see, is this right? Yes, right direction, yes. So in order to figure out how many subclusters, the spread of CBMs, we went through every single subcluster of mycobacteriophages, and we looked to see which subclusters had the CBM of hyaluronate lyase in their minor tail proteins. And after this analysis, we realized that 74% of subclusters contain this. And interesting enough, of the subclusters that don't contain it, 89% are in cluster A. Furthermore, we realized that the protein sequences vary between subclusters, meaning that no two are exactly alike. And this brings us to an argument about conserved domains. Now, with all this information, we'd like to suggest that the CBM of hyaluronate lyase, and when we say CBM, we mean the carbohydrate binding module, should be considered a conserved domain. Now, let's step back and think of what a conserved domain is. It is a distinct functional and or structural unit of a protein that has been conserved during evolution. And if you think of the sheer mass of CBMs across mycobacteriophages, we can think, okay, so there must have been a lot at the beginning, it must have been important, so we have a lot now. Furthermore, what strengthens our argument is how unique it is. And when we say unique, we mean that if we look at the expressor data of 18 CBM-containing minor tail protein genes, the only structural homologous parts are the carbohydrate binding module. Now, we didn't want to just stop there because we're scientists. So we want to think, why does this matter? And this brings us to host range. Because as Connor was saying earlier, so carbohydrate binding module, there's carbohydrates on the bacterial surface. So chances are some kind of variation would equate to some kind of variation. And so in essence, do the amino acid variations in the hyaluronate lyase CBM affect host range due to their differing structures? In order to do this analysis, we needed some data, and Dr. Deborah Jacobs-Serra had posted on the PHAGES forum a bunch of information of a bunch of different hosts and their infectivity across a bunch of different hosts, a bunch of different phages across a bunch of different hosts. And this is a little hard to visualize, so we made a, oh my gosh, we made a visualization using a log graph, and this is really important in doing some espresso analysis.
All right, one of the programs we used is called Espresso. Now, Espresso creates alignments based on structural information. It allowed us to compare two CBM sequences, and it would tell us how similar they were structurally, even if their amino acid sequences were different. With this information, combined with the information from the host range experiment, we were able to check whether there's a correlation between CBM structure and host range. Looking at the data from the host range experiment, you can see that the phages can be divided up into groups based on some infectivity patterns. You look at the first two phages here, cane and DeJager, both infect Gordonia terrae less efficiently than Mycobacterium osteopercanum, and osteopercanum less efficiently than Sphygmatis and Chelone and the rest of these hosts up here. The rest of these phages can be divided up in a similar way until we have 5 distinct infectivity groups. Now, hypothetically, the phages from each group would have structurally similar CBMs if the CBM is in fact a factor in host range. Espresso allowed us to test just this. Essentially what we did is we took each CBM and compared it to each of the others. So for example, we took the CBM from larva and compared it to all the rest of the CBMs. We then ranked them based on how similar they were to larva's CBM. We highlighted the phages that were in the same group as larva, and that kind of showed us how right we were. Ideally, we want red names at the top of the list, because that means the CBM homology coincides with host range. Here's all the data from this experimentation. It's kind of hard to understand, so I'm just going to summarize all the supportive data that we found. We found 3 examples of CBM structural homology coinciding with host range. First example is between groups 1 and 2. I included group 2 here because it's kind of similar to group 1. If you want to know precisely why, I can tell you later. The group 2 phages are in yellow here. And you can see that there are highlighted names towards the top of the list supporting the hypothesis. Next example is pretty great. It's from group 3. Peg-leg CBM is highly homologous to the CBMs of G9C and Fiance, both members of group 3. This is great, but you'll notice that there are red names towards the bottom of this list as well, contradicting the hypothesis. Dang it. So, while this is unfortunate, it is actually possible to attribute these inconsistencies to other factors in host range. Obviously, the CBM isn't going to be the only factor causing different efficiencies of plating on a variety of hosts. So it is possible to attribute these differences to other factors in host range. We finally have this example. This also very clearly supports the hypothesis. Unfortunately, we also have Wintermute, this slacker. Its CBM is very different from the CBMs that it should be matching. It clearly contradicts the hypothesis, which is unfortunate. So the main takeaway I want you to have from this Espresso analysis is that some of the data supports our hypothesis, which is great. Some of it, unfortunately, does not. And we'll talk about why we should be optimistic in this instance a little bit later.
Okay, so we just didn't want to give you numbers and letters. We also want to give you some pictures. In order to do this, we use a program called RASML. Now, what RASML does is it predicts 3D structure. And unfortunately, we are using small little information in a FASTA file. And in order to create 3D structures, you need a bunch of coordinates to show how the protein folds. But we were able to do some converting, and get a flat structure. And it's a little hard to visualize without a model. So here we have the 3D image of hyaluronate lyase versus the flat image of hyaluronate lyase. Now, if I had my computer out, I could take the 3D model and move it around for you. But for this case, and because it's only a picture, we can see kind of how it relates. You see like top here, down, etc. Now, we were able to use this ability to do a little bit of comparison. So the first thing we did is we took BPS, chain 9C, hetero, and larvae, which all have really good infectivity of Gordonia terrae versus Ajuder, which does not have as good infectivity of Gordonia terrae. And we looked at their structures to see where it was the same and where it was different. And the only place that these 4 were homologous were in this little line right here in yellow. And the only place and this happened to be the only place where it wasn't homologous with Ajuder. Now, this wouldn't mean anything if we didn't have more information concerning different hosts, because it could very possibly be that this was the same structure that affects infectivity all across the board. So we did this with M. osteopercanum, Mycobacterium osteopercanum, and then Mycobacterium naturiense, and we learned with phages Geon, Tweedy versus Gaia. And fortunately, you can see that there's a different structure that's homologous between these two that Gaia does not have. When what's really cool is if you take BPS, hetero, and Geon, which affect all 3 very well, you see that they have both of the structures. Now, of course, like Espresso, we didn't have completely supportive information. For example, Charlie and nanosmite had some of the worst infectivity across the board, despite having the features, for example, right here, that should allow them to infect Gordonia terra, and Charlie has a feature that should allow them to allow it to infect the 2 mycobacterium strains. But we can attribute this to structural, not all structures are created equally. So if we look, we see this little tent here in the hyaluronate lice, and it goes all the way down, and this is not evident in neither Charlie nor nanosmite. So we think that this structure is kind of an override feature, and so the lack of it cancels out the other 2 structures, if that makes some sense. And this brings us to our final coincidence or fact thing.
Right, so in our research, both in Espresso and Rasmol, we found some good data and some data that contradicted our hypothesis. This led us to the question of causation versus correlation. Either it's causation, where we're right, hypothesis is correct, or this is merely correlation, where, did I say correlation twice? I don't think so. Or it's correlation, that our hypothesis is, our supportive evidence is merely coincidence, and our hypothesis is incorrect. We have a couple of reasons why we think we should be optimistic here. First, we have a good amount of supporting evidence that would be difficult to disregard as completely random. Second, it theoretically makes sense. Phages have to bind to bacteria somehow, and we know there are carbohydrates on bacterial surfaces. Third, whenever we found a minor tail protein with a carbohydrate binding module, this specific one, we also blasted the surrounding the surrounding genes with HH-pred, and we didn't find any protein matches that matched with anything related to binding. So if it isn't the CBM, we're not sure what it is, although it is possible that we simply haven't characterized this specific structure yet. And finally, there are other factors in host range that would cause discrepancies. All right, we have 3 main conclusions from our experiments. And the first one is that individual traits can be transferred. We saw that Andes had its carbohydrate binding module switched out by a recombinant event, just that one specific attribute. Second, the hyalurate, the CBM should be considered as a conserved domain, seeing how it's been conserved throughout the evolution of bacteriophages. And finally, we think the CBM structure does play a role in host range, and slight variations in that CBM will cause variations in host range.
So people would like to think specifically, for example, the third of our little trio, Nicholas Leonard, and then Dr. Jacob Serra for this host range data. And finally, we'd like to thank you for being here.
Thank you.
Why not? I was clapping for us.
Do we have any questions for these students? Yes.
Wonderful talk. I was curious what you used to compare the modeled protein structures. What program you used? Did you just sort of like visualize them once you had the structure? Sorry, the domain structures, not protein structures. Once you had them modeled.
Is this for Rasmol? Mm-hmm. Oh, yeah. I kind of looked at them in Photoshop and zoomed all the way in. And fortunately, there was literally only that one structure that was unique.
Okay, thank you.
Could we go back to the screen with the carbon binding module lists? Yes. So my question is, do the numbers have anything to do with the order, or is it just the
yeah, the ranked in order. So the number is how similar the modules are to each other. So dandelion, for example, is 81% homologous with larva to CBM.
Oh, okay. Thank you. Yep.
Either one.
All right. I was just wondering if you looked at all at the specific residues of the binding site to see if there's any. I know there's a lot of literature that shows the actual amino acids that are like active in the active site, and if you looked at any homology between.
Unfortunately, we did not. That would be really great to do
we used Swiss model to find the interesting sites on the hyaluronate lye CBM, but we didn't do any analysis beyond that.
Okay. So thank you for the idea.
Hi, I was wondering if you were planning to do like any other experiments like mutagenesis to see if there are other factors involved with infectivity, like looking for those genes and see what happens if you remove some of those genes.
Yeah, it would be fun to cut out Andy's gene 20 and see if it still worked. It probably wouldn't, but that would be. What you could do is you could take a culture of SMEG and strip it of surface carbohydrates, and then if the phage can infect, that'd be pretty interesting evidence.
And this will be future experiments with the wet lab. And unfortunately, we were unable to go back into the wet lab.
Thank you.
But thank you so much.
Thank you. Are there any questions from the other room? Thank you, guys. Great talk.